you. So I'm going to go ahead and start us off. Welcome everyone. My name is Allie Munson. I am a recent graduate from Princeton University, and I will be the moderator for this session today. I wanted to welcome everyone to the fifth Polygence Symposium of Rising Scholars and to the STEM student talks of today. We have a full roster of amazing projects to share with you. And for this session, we have 4 student presenters who are going to give us a talk on a variety of topics. In the room today with us is Judge Alex Winnett, a PhD student at California Institute of Technology, who will be scoring each student's presentation along with myself. Please type your questions in the chat if you have any for each speaker. There is both a chat function and a Q&A function, so you can submit your questions to either, and the way you can find those is all the way to the right. There is a session tab. If you want to click on that, and then you should see sub-tabs for chat and Q&A. So that is where you can submit any questions you might have. And we will allot about 3 to 5 minutes for questions after each presentation. With this, we're going to go ahead and get started with our first presenter, Rachel Wiley, a junior from Cupertino, California. Rachel, let me go ahead and cue you up here.
All right, can everyone see my screen?
Yes.
Oh, sorry, 1 second. All right, awesome. So, hi, my name is Rachel Wiley, and today I'm going to be discussing the chicken Choriallantoic membrane assay for angiogenesis. So, we're all familiar with the classic dissections we had to do in biology class. Whether you dissected a frog, a pig, or scavenged owl pellets for little bones, we can all admit that dissections have been powerful learning tools for students at school. However, few of us can confidently say that these dissections gave us the opportunity to explore, to ask the hows and the whys of human biology. In fact, biology's most troubling questions are hardly addressed in school, leaving students with mere suggestions as to the possibilities of biological studies. Now, if we're stressed the dangers of heart disease, graft rejection, and cancer, shouldn't we be given the chance to explore these issues ourselves? That's where the chicken Choriallantoic membrane, or CAM for short, comes into play. Yes, it does sound like a mouthful, but it's really not as intimidating as it seems. The CAM is one of the earliest anatomical landmarks to develop in the chicken embryo, becoming visible to the naked eye only on day 3 of incubation. Because it develops so early on, it serves as a vehicle for a large portion of embryonic gas and nutrient exchange, making it a very versatile membrane. Given its proximity to the surface, the CAM is very easily accessible through the shell. Both of these qualities make the CAM both useful in modeling a large variety of biological mechanisms, but also simple and cost-efficient to carry out. Research areas that currently involve the CAM model include, but are not limited to, biochemistry, bioengineering, and drug development. However, the most prominent application of the CAM is to model angiogenesis in humans. Angiogenesis, the recruitment of new vasculature, supports the growth and development of cancer. In other words, the more angiogenesis, the more equipped cancers are to grow and survive. The less angiogenesis, the more likely the cancer's nutrient supply will be limited and the cancer will subside. Therefore, strategies to inhibit angiogenesis are highly sought after in cancer therapy research. Because the CAM is highly vascularized and easily accessible, it is an effective tool in the mobilization of angiogenesis research. However, this model is still relatively new and not yet optimized. Most protocols call for cell culture to be transplanted atop the membrane in order to model that particular tissue, but more cost and time-effective models are rarely implemented. Therefore, I sought out ways to make the CAM model more accessible but still effective in modeling angiogenesis, essentially removing the need for cell culture. To find out how, I asked 3 main questions. What are my potential methods, which of them is most efficient, and which is most feasible to do for students at home? First, let's break down the possible methods of the CAM assay. Our first option is the cell culture graft we just discussed. Using a cell culture transplant onto the membrane, the control eggs are compared to those with the experimental variable for changes in angiogenesis. Because this requires the care of cell culture tissue, it requires a lot of time, money, and resources to develop. To bypass the need for cell culture, we can implement a new technique, the limb bud graft. To do this, a limb bud is extracted from one set of chicken embryos and transplanted onto the cam of another set. This way, the growth and development of the transplanted limb placed on top of the membrane can be used as a metric for angiogenesis, along with changes in vascularity. However, this option has little to no research to accompany it, making it a risky choice. To expedite this process even further, we can try using the cam alone. Now we just use the vasculature on the cam itself to assay angiogenesis. This cuts the cost and the complexity by a long shot and cuts time by almost a third, as the maturation of the transplanted cells or limbs is no longer necessary. With that in mind, it's time to test the efficacy of our alternatives. I started experiments first with limb buds. Using fertilized eggs from a local farm, one half are put in the incubator immediately, while the other half are saved to be used later. 4 days later, a viewing window is created by using a syringe to take out excess albumin, the embryonic fluid, and cutting a circle in the shell with small scissors. By now, there should be enough vascular development to check for the viability of the eggs with a heartbeat, as shown in the video here. If the embryo is alive and well, we can proceed with its incubation. And now we can also begin incubating the second batch. 10 days later, the limb buds of batch 1 and the cam of batch 2 are well matured. The second batch is windowed, and a small abrasion to the cam of batch 1 is made to aid in the adhesion of the limb bud graft. A syringe is used to extract the limb bud from batch 1 and carefully placed onto the abrasion. From now to day 25, the limb development is closely monitored, and finally, on day 25, the limb in theory should be developed. This process would be repeated with aspirin to test its ability to model angiogenesis. Aspirin is a known human anti-angiogenic compound, so if vascularity and limb development lessen significantly with aspirin, we can determine that this method is effective in modeling angiogenesis in humans. As for the results, it sadly did not go as planned. While there were some successful attempts as shown, there were so few after so many attempts, time, and resources, it was clear that this was not a feasible route. However, we can still take away the astounding potential of this method. There is really little to no research in this area, so the fact that the cam has the potential to sustain the development of an entirely separate tissue is incredible. With more time and resources, this method could be a great resource for further research. But it was concluded that this method was not ideal for students at home and definitely not the most efficient way to use the cam. So we turned to using the cam alone. On day 4, eggs were windowed and checked for viability. Then the control saline solution or experimental aspirin solution was added atop the cam by saturating antibiogram discs, inserting them, and sealing the window with tape, incubating again. By day 7, the vascularity has had time to sufficiently mature and the cam was extracted. Using small scissors, the cam was gently cut out and cleansed with formaldehyde. A highly controlled camera setup was then used to picture the membrane for further analysis. The results this time were much more promising. Using the images on the right, we can clearly see a difference in vascularity between the saline treated above and the aspirin treated below. Qualitatively, we can see a lower number of recruited blood vessels, smaller blood vessel diameter, and less branching in the aspirin-treated cam. We can use these pictures for some of our quantitative data as well. The control has 6 branch sites on the designated main blood vessel, and the experimental only has 3. For more detailed quantitative data, I am in the process of running the images through a computer program at imagej.net to quantify the decreases in blood vessel diameter, length, and vascular density. I'm also in the process of another trial of eggs, so I have enough data to conduct statistical analysis. This way, we can determine whether or not the differences in experimental and control data are statistically significant. With this data, we can conclude both that the chick Choriallantoic membrane assay is effective in modeling the human angiogenesis response, and that the membrane can be an efficient learning tool for students at home. Also, I'd like to give a special thanks to my mentor, Nina Horowitz with Stanford University, who helped me through this lengthy process. She taught me so much, not only about cancer research, but about the scientific research process and how to ask the right questions. There were so many road bumps along the way, but I'm so grateful to her for guiding me through even when things got murky. And with that, next time you have some free time on your hands, consider testing your experimental cancer therapies on a fresh batch of chicken eggs. Here is a list of my references, and thank you.
Thank you, Rachel. That was great. We're going to open the floor now for questions. If you want to go ahead and type them in the Q&A section or the chat. Okay, I'll start off with kind of a basic one. The work you did was pretty dang cool there. I like how you really wanted to make it very applicable across the board for high school students to be able to do so at home or even maybe, in a high school lab or something like that. I'm just kind of curious, you made kind of the accessibility components quite clear. Was also just kind of curious, is it pretty cost effective as well to implement something like this?
Oh, definitely. I mean, you'd have to go and get some fertilized eggs, but those generally are pretty cheap, especially if you just try and reach out to a local farm. They're generally just as expensive as your normal eggs. But as opposed to the cell culture, which is obviously going to run you a lot more money, you can generally use appliances that you have at home, like small little scissors, or if anything, you only have to get a syringe to extract the albumin and an incubator. But other than that, it's one of the, I think for the potential of the assay, it's worth the small amount of money you have to invest.
Yeah, cool. I was just curious. It
Yeah.
seems pretty unique. We have a question from Alex here asking, how difficult is the process of windowing the eggs? It seems like a very delicate procedure.
Yeah, I thought so too before I first tried it. I was really nervous about breaking the shell, but what you do is you put tape and you extract the albumin by sticking the syringe in the egg. And then once you do that, you can use that point in the egg to carefully cut a circle. And it's honestly, as long as you're going slowly, it's really not that hard. I didn't have issues cracking the eggs or anything. Yeah, it's not as difficult as you'd expect.
Awesome. And one more other question here is, how long did you work on this project?
A really long time, actually. I think it's been around 7 months, just because that first limb bud graft procedure was super hard to carry out because the length was around 3 weeks, so that gave a lot of time for the eggs to, eventually die or just stop growing and developing. So I tried again and again and tried to slightly adjust the procedure to make it more effective, but we ended up just trying another route, and that ended up being a lot faster.
Nice. And then we'll take this one last question here, which is, how difficult would it be for a regular student to manage this in terms of how frequently you checked on the eggs? Maybe speak a little bit to the schedule and how much time it took.
Right. So
No, I think you lost connection there for a second, unless that was just on my end.
I'll just repeat what I said.
Okay,
I'd say the limb bud graft is definitely not as accessible or feasible to do, given time constraints of a student, but the CAM assay only takes around a week, and you only really need to check up on it twice. And just whenever you get the chance, just make sure your eggs are doing well. But yeah, it's super quick and super fun to do. I think it's really feasible, even if you don't have that much time on your hands.
Amazing. Well, thank you so much, Rachel, for your presentation.
Thank you.
Okay, next we are going to move on to Layla. Adeli. I'm sorry also, guys, if I'm butchering your names, please correct me. She is a junior from Pacific Palisades, California, and she'll be going next.
Hi, can you hear me?
Yes.
Okay, for my screen. Share this. 1 second. Can everyone see that all right?
Mm-hmm.
Okay, perfect. So hi everyone, I'm Layla Adeli. I'm going to be giving a presentation on nanotechnology in cancer therapeutics and imaging. So first, a little bit about me. I'm a junior at Palisades Charter High School. I play the violin in the LA Youth Orchestra, and I also run cross country and track as a distance athlete. So first, a little roadmap. Today I'm just going to be talking about what nanotechnology is, also what is cancer, and then I'm going to go in a little bit of different types of cancer imaging and treatment just for background. And then I'll also be giving some examples of nanotechnology in cancer imaging and treatment. And then at the end, I have a little proposal of a novel therapeutic based off of what I learned reading studies. So first, what is nanotechnology? So it basically is just the manipulation of matter at the nanoscale for industrial purposes. So the nanoscale is very, very small, as you can see in this diagram right here. It basically goes from the size of a glucose molecule to the size of a virus. So we're talking about manipulating atoms and molecules. And so in biology, we can use nanoparticles like liposomes and quantum dots and carbon dots. And we can even manipulate DNA to make origami for drug delivery, which is really cool. And basically liposomes, quantum dots, and carbon dots, I just named those because they're bioavailable and also very biocompatible. So you can use them with different types of like chemotherapeutic drugs and things like that without actually affecting the drugs themselves, and also without affecting human beings or whatever organism you're using them in. So second, what is cancer? So cancer is uncontrolled cell growth. It comes in many different diverse forms and tissues. And for that reason, it can be really difficult to image and treat because obviously human bodies are all so different from each other. And if we only have one way to image or only a couple ways to treat so many different types of cancer, it can be very difficult. And so cancer is just named by the type of cell that will cause the tumor, like the physical appearance or the phenotype of those cells, and then how advanced the disease is. And this is just a little diagram of the hallmarks of cancer, how you can tell if it is cancer or not. So next up, current cancer therapies and detection can include MRI, CAT scans, X-rays. These aren't really super specific to cancer themselves, but they are currently some of the most helpful ways in determining if there's a tumor or not. And then you can use a biopsy to see if you have a mass, if it really is cancerous or if it's benign. And then currently there are 5 pillars of cancer therapy. Those are immunotherapy, chemotherapy, radiotherapy, molecular therapies, and surgery. So chemotherapy and radiotherapy are pretty toxic. So there's lots of room for improvement there. Immunotherapy is pretty new. It just harnesses your immune system to be stronger to fight against the cancer. And then surgery is obviously trying to cut out the tumor, things like that. Molecular therapies are just medications. And so we now have this potential for a 6th pillar, which would be hopefully nanotechnology. So first, a little example. Nanotechnology, at least when I was learning about it, for me, it was very difficult to really grasp the concept just because it's so small. But for cancer imaging, what we can do is we can combine a targeting agent, like an antibody, which will just like target an antigen that can be found on certain cancer cells. And then you can also use an imaging agent that can improve the quality of the images. So if you have a more dense tissue type, then that won't get in the way of an MRI image. So there's this one example that I thought really highlighted this very well. So there are these antigens that are classified as BRCA1, and they're really found on breast cancer cells, but they're also found on gastric cancer cells. So gastric cancer is stomach cancer. And because these antigens are found on those cancer cells and only those cancer cells, you can use an antibody, which is specifically targeted to this type of antigen. And if you conjugate that, you combine it with a fluorescent nanoparticle, then you can use it in gastric cancer imaging. So first you target the BRCA1 antigen with this specific antibody, and so that will precisely locate the cancer cells. And then if you use a fluorescent nanoparticle, or FMNP, then you can enhance the fluorescence of the imaging and make it even clearer if there is something truly there. And this is just the study that I found that in. So now in cancer therapy now, it's really important to note that these are pretty toxic, they're non-targeted, and especially in chemotherapy, there's a really poor bioavailability in those types of drugs. So there's not a lot that we can use. But with nanotechnology, hopefully the goal is to really enhance older therapies with targeting agents, like I said before, antibodies and ligands. Ligands are just molecules that you can also find on these cells. And this will hopefully lower toxicity, because then the drugs will only go to the cancer cells and not everywhere in the body. And you can use safe and biocompatible nanoparticles for drug delivery. Like I said before, carbon dots and liposomes are really great examples of those. And over here is just some examples of certain types of targeting agents that you can use. You can use half of an antibody, a full antibody, anything really works, and that can just target whatever cell you are trying to target. So now I have an example of nanotechnology in cancer therapy. And this is specific to chemotherapeutics. Like I said before, chemotherapy has a lot of room for improvement. So this is a diagram that will be helpful in 1 second. So there's a recent study that showed that if you use a gadolinium encapsulated carbon dot in a combination photothermal chemotherapeutic drug delivery vehicle for triple negative breast cancer, that would be very, very effective. So gadolinium is an element. It's pretty toxic if it's free in the human body, but if you encapsulate it in a nanoparticle like a carbon dot, which is biocompatible and bioavailable, then it won't be toxic anymore. However, gadolinium is really great because it can enhance MRI images. So it can make it very obvious where the tumor or the mass is. And then what you can do is you can also add a chemotherapy drug, like doxorubicin is used heavily in breast cancer, and a photothermal agent. So then you can use radiation to also kill cancer cells, and you put this all in one carbon dot platform. And if you inject this, in this study they used mice, so they injected it into the tumor of the mice, then you can find that MRI results are clear. And there's also no evidence of doxorubicin, so chemotherapy-related toxicity, to the organs of the mice used, which was previously a really big issue. And then also, by adding radiation, you can also suppress tumor growth. So overall, it's showing that nanotechnology, while it might not be a pillar of cancer therapy on its own, it can really help the cancer therapies we have today. So next, this is my proposed novel, therapeutic, but also diagnostic. So what I did is I took that last study that I just showed you, and I wanted to see how can we improve this more. So you'll see a lot of this is very similar to the previous slide. Just some important things to know. NGALs stand for neutrophil gelatinase associated lipocalin. All you really need to know in this case is that it's expressed on early grade ovarian tumor cells, but not on healthy ovarian cells. So it can be used as an ovarian cancer marker. but it's also expressed on neutrophils. So if you're going to use it for cancer therapy, you're going to also want to use another type of molecule ligand, in this case, a receptor. It's called PDGFR alpha. And that's expressed on ovarian tumor cells, but not neutrophils. So this really adds another level of specificity when trying to target just ovarian tumor cells, so that chemotherapy or a photothermal agent will only enter those cells and nothing else. So like I said before, you combine the gadolinium encapsulated carbon dots with some sort of NGAL binder, for example, MMP9, and PDGFR alpha, and this will act as an MRI enhancer because gadolinium will improve the images that MRIs provide. So those are T1 images. It's also a chemotherapeutic delivery vehicle. You can also use doxorubicin in this case, and as phototherapy potential with the photothermal agent. So this creates a combination therapy. So overall, this can improve chemotherapy and MRI imaging and can be used as a diagnostic and a therapy. So thank you so much. I want to acknowledge my mentor, Nina Horowitz. She's a PhD candidate at Stanford University, and even though she has a very research-filled schedule, she still guided me in this really complex world of nanotherapeutics and nanotechnology. Cancer therapeutics and nanotechnology. And thank you.
Nice job. We're going to open up the floor now to questions. Yes, okay, there is one. So how well do nanoconjugated treatments penetrate deep into solid tumors?
So in this case, all of the nanotechnology therapies that I found, all the studies that I found, were only related to mice. None of these have reached human level clinical trials, as it's a really, really new form of cancer therapy. A lot of the studies I found were only from 2021. So these conjugated treatments, obviously they use them so they could inject them into the tumors. While that might not work as well in a human being, because obviously we are a lot bigger than mice, and so a needle would not penetrate through our entire body or into a tumor that's very deep in our tissue. In this case, it worked out very well because obviously they were allowed to inject the tumor directly. But theoretically, with targeting agents, it should penetrate very deep because you're only going to that one cell, and by having receptors and aptamers interact, the drugs or chemotherapy drugs or whatever agents you're trying to use, in theory, should reach those tumors directly.
Gotcha. And a follow-up to that question was, what ideas do you have to improve or optimize nanoparticle delivery to the center of solid tumors?
So I think in my proposal, I think that's probably the extent to my idea, just adding more targeting agents, just so you can make sure that it's actually reaching those cells. I didn't read anything about the therapies or the nanoparticles not being able to actually penetrate the tumors regardless. So I'm assuming that by using these receptors, it kind of allows the cancer cells to just open up to, or the tumor microenvironment to open up to these nanoparticles regardless. It's kind of like a bit of a trick. And yeah. So.
Alrighty, amazing. I think with that, you got a few congratulations in the chat here. Let's see if there's one more question. Yes, okay. Is this the line of research you would like to continue?
Definitely. I've always been very interested in cancer therapy. And so when I started this project with Nina and we started learning more about, how could we improve cancer therapy, and she showed me this journal of biotechnology, I was in awe that I had never heard of this, and also how amazing it was that we could now make chemotherapy, and we can improve all these older therapies instead of having to just start all over again. And I definitely think that this has so much potential to help so many people, just because it's so customizable to different types of cancers and different people and the different grades and levels of cancer. And it's definitely something I would like to pursue in the future.
Thank you so much.
Mm-hmm.
Okay, so I think with this, we are now going to move on to, I'm not sure if Kabir is joining us today in the end, so I think we'll go ahead and move on to Tarun Rao, a senior from San Jose, California. Oh, so sorry, Kabir is here. Kabir, can you, on the top right, you should see a button that says ask to share audio, and yeah, I'll remove you here, sorry. And then, sorry, Tarun, if you could also press the button up in the top right that says share audio and video, it will put you back in the queue. Okay, great, thanks. Okay, perfect. Sorry about that, Kabir. Kabir Ramzan is a sophomore from Saratoga, California, who will be presenting for us next.
All right. Can you all see my slides?
Yes.
All right, sounds good. Okay, so today I'm going to be talking about TogaTech Core, which is an open source framework that I've been creating for rapidly and easily building end-to-end encrypted, privacy-centric, and interoperable applications. And so kind of what that means, we have end-to-end encryption, which is just that when you have two users that are sending messages back and forth or sending some sort of data, those users are the only people that can access that data and not the company that runs the application. This is really important for privacy reasons as well, to make sure that your data is safe and secure and is owned by you and not by the company. And then the interoperability aspect is that using this framework, different end-to-end encrypted apps can communicate with each other to share data between them using end-to-end encryption and privacy. So there are a few end-to-end encrypted apps that you've probably heard about, like Signal, iMessages, and Telegram. These are each kind of the standalone end-to-end encrypted messaging platforms. However, they have a lot of focus on actually building out a lot of the cryptography, and so can't add more features onto it, like something like Slack, where there's a lot of different channels and threads. And also, they don't really talk to each other. So those are some problems that I'm trying to fix using this core framework. So what is cryptography? I can start off with a quote here. Cryptography is about communication in the presence of adversaries. Kind of what that means is that if you have two people that are communicating, there might be other people that want to see that communication. So you want to make sure that communication is, one, it's encrypted so that only the two people can read that message. And second, you want to prove that it came from you. Otherwise, these adversaries can just pretend and make up messages to pretend that they're you. So those are two very important components. And so as part of this cryptography presentation, I'm going to be going over a few key concepts and then how they apply to the TogaTech core framework. So first off is symmetric encryption. This is pretty straightforward. Basically, you have one key, which is kind of like a password. It could be used to encrypt and decrypt some sort of text. So as you see here, it encrypts the text and then decrypts with the same key. One example you may have heard of symmetric encryption is the Caesar cipher. So in this case, in the Caesar cipher, the key is a number between 1 and 25, and that's how many letters you shift in the alphabet to like encrypt something, and then to decrypt, you shift all the letters back. Although, you know, this is only 25 numbers, so you can try all 25 possibilities to get the correct message. we use something called the Advanced Encryption Standard, which just kind of has longer keys and has other protections against like trying all the possibilities. And then we have asymmetric encryption, which is a very similar concept, except instead of one key, there are two keys, as you can see here. One of them is the public key, and the public key is used to encrypt text and is known by everyone because it's public. And the private key is known only by you and is used to decrypt the messages. This way, if you want, you can give your public key out to anyone and they can send messages to you that only you can decrypt. Otherwise, if you're using symmetric encryption and you give that one key out, it can be used for both encrypting and decrypting, which means other people can decrypt your messages, which is probably not something that you want. So here's a little diagram of how that works. If the green user wants to send an encrypted message to the blue user, they use the public key to encrypt it, and the blue user can use their private key to decrypt that message. There's some libraries that do this, such as PGP, which uses an implementation of an algorithm called RSA using prime numbers. We can go over that more in the Q&A if you're interested. And then there's also SALT, or the networking cryptography library, which uses elliptic curves instead. And this is the one that we mainly use in TogaTech Core. However, we do have support for both. And then there's one key limitation in this, that the size of the message has to be smaller than the size of the keys. And also when the message gets bigger, it becomes a lot slower to encrypt and decrypt that message. So what we tend to do is use asymmetric and symmetric cryptography at the same time. So we create a message, and then we create a symmetric key to encrypt that message, and then we have an asymmetric key to encrypt that key. So it's just kind of a second layer. And then hashing is basically just a one-way function. You have an input and then it brings it to an output. And when you have that output, you can't get back the input very easily. Also, if you just change like one small thing in the input, it completely changes the output. And it's also a good compressor as well, because this hash is always going to be the same length, no matter how big the message is. There are a few hashing algorithms that are supported by TogaTech Core. The ones where I marked with broken, that just means that you can have two inputs that come to the same output very easily. But other than that, SHA-256 is the one that's probably most commonly used, along with SHA-512, which is also pretty commonly used. And then digital signatures is the last concept in cryptography I want to talk about. It's basically just the asymmetric encryption, but in reverse. So originally, how I said you can use the public key to encrypt because everyone knows the public key, and only the private key to decrypt. Well, the way they're mathematically linked, you can actually do it the other way around. And how this would work is if the green user wants to prove that they're sending a message, they can encrypt it with their private key and then send it to the world, and anyone can decrypt it with the public key. But the only way it could have been decrypted with that public key is if it had been encrypted with the private key in the first place, proving that you had that private key. And then some limitations, again, it has to be smaller than the size of the keys, the size of the message. And also, you don't want to be encrypting your message using this method because anyone can read that message. So instead, we'll just do the same process, except use the hash of the message instead. And then the person who gets that message can compare the hash. So just to recap, symmetric encryption is one key, asymmetric is two keys. We have hashing, which is a one-way function, and digital signatures, which is asymmetric encryption in reverse using hashing. And then the TogaTech Core consists of our Tenvoy cryptography library, which includes all of those features, as well as a random number generator for randomly generating these keys, and a key generator, which uses a username and password to do the key generation. We have the TogaTech Auth and API, which is our authentication layer. We have TogaTech encrypted capsules, or TeX for short, which is our real-time encrypted data storage. And then we have the TogaTech SDK to access all of these core features through a simple programming interface. And then TogaTech Spark is our first major application built on top of TogaTech Core. It's a fun, feature-rich, and end-to-end encrypted collaborative chat platform, sort of like Slack. Everything is signed and encrypted for that extra security. We have support for direct group chats, typing statuses, currently working on reactions, profiles, and in the roadmap, we're going to be creating these spaces with channels, threads, roles, and more. It's kind of a preview of what we're working on here. And we have TogaTech Auth, which is sort of like something sort of like sign in with Google, except it has a few extra security features. You can sign to any app using TogaTech Core with the same username and password, which allows these apps to kind of talk to each other. You can give permissions for individual apps, and then asymmetrical keys are generated through an algorithm to prevent brute forcing of the password. And then we have capsules, which is our encrypted data structure. We have the ID and the name. These are just used to kind of identify that capsule. We have a header, which includes all those encrypted symmetric keys. Remember that two-step process we have to do for asymmetric encryption includes like ranks and permissions, like who can read and write to that capsule. We have the actual body, which contains that encrypted message, who last edited the capsule, along with their digital signature and hash. And then we have chains, which we'll get into in a few minutes. This is what a capsule might look like for TogaTech Spark. We have like a message, And then in the message, we have like all the different roles and keys, and then we have who sent it, the signature, and the data. And then the actual, maybe there's a chain of reactions, like all the emojis that are linked back to this message. And you can see, like, even though this is the data of the message, there's all of this other stuff that has to go into it first. And then chains are basically just a list of capsules that link back to an original capsule. So you can see here an example, you might have a direct message, and then off of that direct message, there's a list of all the messages that link back into that like grouped chat. So these capsules and chains are stored persistently in encrypted form on the server, synced in real time with anyone listening to these capsules. You can scan for them by the name or rank, like give me all the messages. You can directly get the capsule from that ID, or you can access chain elements by knowing that like header capsule. So for example, if you can say, give me all the direct messages, it'll give a list of the direct messages. And when the user clicks on one of them, you can say, give me all the messages that are linked to this direct message group. And then finally, we have transmissions. Transmissions are just like taking a capsule and kind of making a smaller version of it. So, for example, if you're having a phone call that you want to be end-to-end encrypted, you don't need people to be like editing that phone call or seeing it after it happens. You don't need it to be stored permanently. So you can kind of take away all of that extra features stuff that we had put previously. So yeah, it's not stored persistently on the server. It is sent in real time though, directly to the user, but there's no like editing or any of that. So that kind of summarizes TogaTech Core with our cryptography library, authentication, data storage, and the SDK to access all of these features. I also want to give a special thank you to my mentor, Nick, who has helped me throughout the past spring, summer, and early fall to be building this core framework and the end-to-end encrypted chat platform, help me out with the bugs and bouncing ideas and features, all that. So thank you very much for listening to this presentation. And please let me know if you have any questions.
Thank you. That was very impressive. We'll open the floor now for questions.
I think I see one from Alex.
Yeah. Oh my gosh, I had the same question, which was, how did you come up with the name?
So I can start with TogaTech. So I think as you introduced me that I'm from Saratoga. So that's where the Toga part comes in. And also its origins in like the Roman Empire being like the basis for modern civilization. Kind of how this is like a framework that can be the basis for a wide variety of end-to-end encrypted apps that can be built on top of it. And then also just how from Rome we have Latin, which is the basis of a lot of modern languages, just like this is kind of securing modern communications. And then core is like, it's like a kind of at the core level, like the inner workings of all the applications. For example, with TogaTech Spark, it uses that core framework, that chat application uses the core framework. The core framework does like a lot of the heavy lifting. So the demo that not the demo, but the picture that I showed was written in probably around under 500 lines of code, just because it's using all the core features, which do most of the work for you already. So it's kind of like at the core of everything.
Nice, very efficient. Another question here was just, for how long did you work on this project?
Probably, well, I started working on the authentication and cryptography library maybe 1 or 2 years ago, and then been working on the core framework and the chat application for the past maybe a little over half a year, probably since April, when I started working with my Polygence mentor.
Very nice. And another one asked, how do you decrypt the encrypted data stored on the server?
Right. Okay. So basically how this works is it uses that asymmetric encryption. So you have those two keys. And the public key is used to encrypt the data. So when you send a message, before it gets sent to the server, it gets encrypted. That way no one else can read it. And then it gets stored on the server. And then for the person who's receiving the message, they can kind of request it from the server, and then it'll become in this encrypted form. And that also plays into the authentication to make sure that they have the permission to request that data. And then when they get that data on their computer, the core framework will automatically decrypt it using the private key. And that's kind of how that link works.
Nice. And last question we'll take was, what was the most unexpected or surprising finding from this project for you?
Right. So one of the things is, I've been doing some research into like any other like applications that do this end-to-end encryption. And one of the surprising things that I found is that these applications all kind of sort of reinvent the wheel. Like they have their own cryptography setup. They've got their own sort of isolated ecosystem. And I couldn't really find anything where you can have like multiple different applications that can talk to each other. Like you can share files between applications that are end-to-end encrypted, or really like some sort of base framework to build everything on top of, which is kind of why I started building this in the first place.
Amazing. Thank you so much. We're going to move on then to our next presenter, which will be Tarun Rao, a senior from San Jose, California.
Can everybody hear me?
Mm-hmm.
Okay, I will now share my screen. All right, can everybody see the screen, and can everybody see the laser pointer?
Yes, I can.
Okay. So, hello everyone, my name is Tarun Rao, and today I will be discussing the intricacies of bioprinting vasculature. So I am from San Jose, California, and currently I'm a senior at Leland High School. In college, I plan to major in biomedical engineering. For my Polygence project, I wrote a review paper detailing the process behind bioprinting, and in the end, I gave my opinion on what I thought the best bioprinting method was. Some of my hobbies include playing tennis and playing the violin. Before we begin, I would like to introduce some key terms that will be essential when comprehending the significance of bioprinting. The two most important terms that you should remember are bioink and cell viability. A bioink is simply a cell encapsulating material that is printed along with the cells, and cell viability refers to the proportion of live and healthy cells within a population, and it demonstrates how effective a bioprinting method is. I'm sure that you all are familiar with the concept of an organ donor or a donor list. Every year, 8,000 people die because of the imbalance between organ donors and patients, and also because organs can reject their patients. To combat this problem, researchers have engineered in vitro organs, or organs developed outside the body, for this purpose. However, these organs are very small in size because they lack the functional vasculature needed to transport nutrients. Take, for instance, this heart right here. This heart was a bioprinted heart, and it is meant for a human, yet it is only the size of a rabbit's heart. As clearly stated, it lacks the vasculature needed to transport the nutrients without limiting its size. So bioprinting has the potential to create this functional vasculature, which can be implanted into these organs, which will greatly increase their functionality. So what is bioprinting? Like 3D printing, bioprinting is an additive manufacturing technique in which layers upon layers of materials are stacked on top of each other to form a 3D structure. Unlike 3D printing, bioprinting uses cells to form a tissue structure instead of plastic or metals. The two overarching categories of bioprinting are scaffold and scaffold-free bioprinting. In the image on the left, we can see scaffold bioprinting, as scaffold bioprinting uses a bioink and the cells proliferate after they have been printed. The scaffold here is shown in gray, which represents the support material. And contrary to that, scaffold-free bioprinting allows the cells to first form aggregates or clusters, and they are then printed in pellets, shown by the spheres over here. From there, they fuse together, forming a 3D structure. To understand the significance of bioprinting, a brief introduction to the vascular system is necessary. Our vascular system is incredibly complex and cannot be replicated simply by stuffing a tube into an organ. As you know, there are three types of blood vessels: arteries, veins, and capillaries. Capillaries remain the most simple type of blood vessel because they are composed of only two layers of cells, epithelial and endothelial cells. Contrary to that, arteries and veins are both composed of three distinct layers of tissue shown here: the tunica externa, the tunica media, and the tunica intima. Because of the simplicity of capillaries, many bioprinting methods revolve around printing them instead of arteries and veins. And now we shall have a look at three of these methods. The first bioprinting method I will be discussing is extrusion-based bioprinting, in which the bioink is extruded in a continuous filament. The three sub-methods of extrusion-based bioprinting are pneumatic, piston, and screw-based, as shown in the image on the top right. In pneumatic-based bioprinting, the ink is extruded through the use of air pressure, decreasing the volume in the bioink chamber. Piston-based bioprinting operates very similar to how a syringe would work, in which case mechanical pressure is applied, forcing the bioink out. Finally, screw-based bioprinting operates by placing the bioink into a screw, which is then slowly twisted, forcing the bioink out. The main selling point behind extrusion-based bioprinting is its high variety of bioinks, which includes substances such as hydrogels, tissue spheroids, and decellularized extracellular matrix. However, a big issue that remains with extrusion-based bioprinting is its very low cell viability, which ranges between 40 and 85%. The reason for this low cell viability is that when the bioink is being extruded out of the nozzle, it undergoes immense pressure, causing mechanical damage and structural damage to the cells. To combat this issue, shear thinning bioinks have been developed, they are bioinks that are able to decrease their viscosity or thickness, allowing them to flow out of the nozzle much more easily, keeping the cell structure intact. Additionally, extrusion-based bioprinting can have a low geometric complexity because the weight of the individual layers can collapse in on itself. To solve this problem, researchers at Carnegie Mellon University have developed a new bioprinting method called the free-form reversible embedding of suspended hydrogels, also known as FRESH. In FRESH bioprinting, which is shown here, any structure can be formed as a primary hydrogel is printed into a secondary bath, and then this secondary bath holds the primary gel in place until it's melted, and this eliminates the problem of low geometric complexity. Droplet-based bioprinting is another prominently used bioprinting method, but instead of the bioink being deposited continuously, it is instead deposited in droplets as shown here. After being deposited in droplets, the droplets then fuse together, and then layers upon layers are stacked on top of each other, and finally they are subject to post-curing processes, forming a 3D structure. The most common droplet-based bioprinting method is drop-on-demand bioprinting, in which the droplets are deposited only when necessary. The three sub-methods of drop-on-demand bioprinting are piezoelectric, thermal, and electrostatic bioprinting, each of which uses different methods to generate droplets. Due to the prospect of the nozzle getting clogged up, droplet-based bioprinting is limited to just low viscosity bioinks. However, many of the same hydrogels that are used in extrusion-based bioprinting are applicable for use so long as the extracellular matrix content is decreased. Cell viability for droplet-based bioprinting can reach up to 90%, but is often less than 85%, and this method has the lowest cost out of the three methods I'll be discussing. Our final method that I'll be discussing is stereolithography, which uses a laser. Looking at this image here, a laser source is directed towards a mirror, which then reflects said laser into a vat of photocurable resin. Once the light touches the resin, it then induces cross-linking, forming a hard layer. There, an elevator platform lowers the completed layers, and a recoater blade applies a fresh layer of resin to be hardened, forming a 3D structure. The common source of light for stereolithography tends to be ultraviolet light. However, this light is very damaging for cells, and an alternate solution is visible light bioprinting, which has formed viable structures. The bioinks for stereolithography tend to be variants of those used in extrusion and droplet-based bioprinting, but they have been chemically modified by adding a photoinitiator to make them reactive to light and thus forming layers. A minor disadvantage of stereolithography is a long post-processing time, but the results are well worth the wait, as stereolithography has the highest cell viability out of all the methods, along with the highest resolution, and it has unique advantages of no cell density or the number of cells within the bioink or viscosity restriction. To conclude, why bioprinting? There are many other methods of generating functional vasculature, such as cell sheet engineering, electrospinning, and mold casting. However, these methods all require additional substrates. They have a much more complicated manufacturing process, and they have to create a limited structure. So really, these methods cost more, take more time, and are less versatile than bioprinting. The biggest advantage for bioprinting is that it can generate a 3D structure with no preset regulation on the shape, while also being able to control the cells within that shape. This means that it will induce proliferation within the structure while also making sure the cells don't grow out of control. Now that we know that bioprinting is the best method for generating functional vasculature, what is the best bioprinting method? I believe that stereolithography holds the most potential for creating functional vasculature, given its high resolution and cell viability, along with its unique advantages of no bioink viscosity limit or cell density restriction. Right now, the challenge for bioprinting remains creating vasculature with multiple layers simultaneously forming veins and arteries. Bioprinting is the future, and I truly hope that those who have suffered industrial accidents and veterans who have served their country honorably will be able to live fulfilling lives with bioprinted appendages. I would like to take a moment to thank my mentor, Nikit Calcante, a second-year PhD student at UT Austin. He introduced me to the idea of academic research and really helped increase my knowledge in regenerative medicine. Thank you so much, Nikit. Thank you for listening to my presentation. Any questions?
Thank you. Great job. give everybody a second to digest that, and here we go. Is bioprinting used in real-life application today?
Yes, bioprinting has been used in real-life applications, as shown in the first slide. A bioprinted heart was created. However, now bioprinting revolves around making the vasculature necessary, and this vasculature has actually been implanted into many created organs, such as bladders, livers, and kidneys.
Nice. And just for time's sake, because I realize we've kind of gone a little bit over maybe, and we have one more. Any other questions, just put in the chat and Tarun can answer them directly. And we're going to thank you, Tarun, for your presentation and for your answers. We're going to move on to Tanisha, I believe. Wait, let me see this go. Yes, Tanisha Mehta, a sophomore from San Jose, California. Yes.
Okay. Hi, everyone. I hope you're having a wonderful time at the symposium this morning. Today, I will be presenting to you a project that is very important to me and very close to my heart, my COVID-19 mental health blog. So let's start off with some introductions. Who am I? I'm Tanisha Mehta. I'm a sophomore at Bassist Independent Silicon Valley, and I absolutely love medicine and biology. Medicine is something that I truly see myself working in someday. I'm extremely passionate about mental health awareness, activism, and advocacy, hence this blog. And some things about me, I do speech and debate, tennis, and I play piano. And here's just a cute picture of my dog, because who doesn't love puppies? So what is my project? So how many of you have interacted with a person suffering from a mental illness? How many of you have a friend, family member, or acquaintance who suffer from a mental illness? The number of people suffering from a mental illness or disorder is increasing drastically day by day, which is why it's even more important for mental health awareness and advocacy for every person. So this project is something that I wanted to create for a while now. And with Polygence, I was able to turn my vision and my dream into reality. So under the guidance of my mentor, I was able to create a COVID-19 mental health blog for both parents and children that spreads awareness and highlights the importance of mental health of children during the COVID-19 pandemic and provides practical ways that parents can support their children. So essentially, this was a long, extensive process of collecting dozens of research papers, combing through them, and finding details and studies that would truly be helpful and necessary to create my blog. So the big takeaway, the thing that I want every single person who's reading my blog to learn, is that we should all make an effort to embrace mental health as something that is just as critical as physical health. So now my blog, I'm just going to take a second and I'm going to paste the link to my blog in the chat. And I would truly appreciate it. Oh, sorry about that. I would truly appreciate it if you guys would take a look at the blog and just read along with me. So I'd appreciate if you guys would navigate to the first section, which is what is COVID-19? So according to the World Health Organization, COVID-19 is an infectious disease caused by a newly discovered coronavirus. Most people infected with the COVID-19 virus experience mild to moderate respiratory illness and recover without requiring special treatment. But please don't make the mistake of underestimating this virus, because since the start of the pandemic, there have been a devastating number of over 4 million deaths and climbing. So if you guys will navigate to the next section about mental health. What is mental health and why is it so important? So when we think about mental health, we almost always default to thinking about physical health. And mental health is simply how we feel about ourselves, our life, our family, our friends, and the world around us. If we're stressed or depressed, our poor mental health can impact our physical health. And studies have shown that while in a depressive state, people are often distracted from their physical well-being as well. So you might be wondering why I have two giant semicolons on the right. So these semicolons essentially symbolize a continuation of mental health awareness and support for those dealing with a mental illness or loss of someone from suicide. In 2019, suicide was the second leading cause of death among children and adolescents ages 13 to 19, and the leading cause of death among 13-year-olds. So why is mental health taboo? Mental health is often stigmatized and devalued, maybe due to a lack of understanding and fear. Mental health disorders and illnesses are commonly perceived to be abnormal and out of the ordinary. So people might have religious indifferences, cultural mindsets that prompt them to kind of feel a certain way about mental health, or feel a certain way about people who suffer from mental illnesses or disorders. And some people might even have opposition against therapy or meds used to treat people with mental illnesses or disorders. So now please move on to the next section in my blog. What does COVID have to do with mental health? As COVID-19 took precedence over all areas of our lives, less resources and attention was devoted to mental health services. So combine this with the physical ramifications of the virus, things like school closures, economic concerns, and social isolation, and this all contributed to a drastic decline in mental health among children. So now we're going to move on to the next section of my blog, COVID-19's impact on children's mental health. Even in normal circumstances, dealing with mental health as a child is exceptionally hard. You know, self-awareness of one's mental health doesn't come easily either. It's very hard for many kids to first understand what mental health is, what mental health disorders are, and then actually realize that, hey, I'm actually suffering from a mental illness or a mental health disorder, or my friend is suffering from a mental health illness or disorder. And so my blog discusses COVID-19 and its impacts on the mental health of children. It's divided into 5 key sections: screen time, child abuse, school closures, economic concerns, and social isolation. So what each section does is that it talks about COVID-19 and how it's impacted things like screen time, maybe it's increased or decreased them, and how that in turn impacts children's mental health. And the blog also includes a section on children with pre-existing conditions or special needs. So now we're going to move on to the section on what parents and guardians can do. So it's divided into 4 key sections: the parent-child relationship, family routines, open communication, and some tips for parents. So whether you're a parent, guardian, or caregiver, you play a significant role in your child's life. As a result, you have immense influence on the mental health and well-being of your child. So in the blog, I discuss the importance of the parent-child relationship, family routines, and open communication between parents and guardians and their child. So the blog also contains really useful tips for parents taken from a research paper concerned about their child's mental health during this pandemic. So please, if you're a parent or guardian and you're truly concerned about your child's mental health during this troubling and difficult time, please take a look at these tips, and they truly are very useful. So now for some acknowledgements. I would really like to thank my amazing mentor, Emma, who is currently at Columbia University, for supporting me, guiding me, and always believing in me and my vision throughout this project. I would also like to thank Polygence for giving me this wonderful opportunity to create a project I am immensely proud of and that I can use to better my community. Thank you. And I wanted to quickly close out with sharing with you guys some analytics of my blog. So I launched the blog a little under 2 weeks ago, and as of now, I have 350 users and counting. And so please, if you truly believe in mental health awareness and activism, please take a look at the blog, please read through it, and share it with your family and your friends. And if you guys are interested in some references, I also have a bibliography in my blog as well. Thank you so much for listening.
Thank you, Tanisha. It was a really great presentation. We will open it up quick for Q&A. And then I'm going to have to hop off and go moderate another session. I guess I just will start off with one quick general one. How long have you been working on this project and developing your blog?
Yeah, so I've wanted to do this for around a year, I think, and this year with under the guidance of my mentor, I've been working on this for around maybe 4.5 to 5 months. So the whole of summer and then a little bit of the school year as well.
Very nice. And we'll go for one more here from Alex. Have you seen anything about the impact of mask wearing on children's social development or mental health?
No, I don't think I've seen any research papers that have talked about that either, but I do think that masks are, like, given the conditions, masks are very important to stopping the spread of COVID-19 and protecting yourself, especially for younger children who are not vaccinated. And, if you don't wear a mask, I think that might increase a sense of fear in children's minds, which can sort of indirectly impact their mental health as well.
Amazing. Well, thank you so much again, everybody,
Thank you.
for joining us today. And thank you, Tanisha, for that presentation. You guys have all worked so hard on your projects, and a huge thanks also to the audience members for coming to support these student presentations. We at Polygence are incredibly proud of all your achievements, and I gotta say, I'm thoroughly impressed by what you guys have accomplished. And we're just really glad to cheer you on and celebrate you on the stage today. So we would like to encourage you to attend and support other student presentations throughout the rest of the day. And you can kind of just go over to the reception area and click on other sessions and browse. But thank you so much again, everybody, and great job today.